So the project that I will present today is a really simple project where we just use basic technique to validate or to test the specificity of different antibodies. Nevertheless, it took us nearly 4 years to publish this data. And I will tell you at the end why. First of all, I would like to give a bit of a background about the molecule of interest. So we are studying the Duffy antigen receptor for chemokine, or in short, DARC. So that has been the name of the molecule of this receptor for a long time, and more recently it was renamed SEKR1, which stands for atypical chemokine receptor 1. So you may have heard already about chemokine and the role in cell migration. So briefly, when a chemokine binds to the receptor, which is a GPCR, that will trigger a different signaling pathway, which ultimately leads to the migration of the cell. And in contrast, the SEKR1, and despite the fact that it binds several chemokines, does not signal. So as a consequence, there is no cell migration. And instead, SEKR1 regulates the level of chemokine in the tissue. So that's why it's an important molecule. And SEKR1 was ascribed a unique expression profile, as it was shown that it's expressed only by red blood cell, venular endothelial cell, and some neuron in the cerebellum. And over the past year, several antibodies have been developed to map the expression of SEKR1. But in the chemokine field, 2 antibodies are widely accepted and used. And these antibodies are the following ones. So the clone 2C3, which recognize the human SEKR1. And more precisely, it recognizes an epitope of 5 amino acids in the N-terminal of the receptor. And there is also the 6B7 clone, which recognizes the mouse SEKR1 in its native conformation. And both these antibodies help to confirm that SEKR1 is indeed expressed only and highly by red blood cell, venular endothelial cell, and neurons. And despite that, there was a study published by Hu et al., which challenged this concept. And they propose that SEKR1 is actually expressed by macrophages and monocytes. And also, the data suggested that SEKR1 is not expressed by erythroid cell or venular endothelial cell. And the main issue from this study is it relies mainly on commercially available antibody, which have never been validated. So we decided to test this antibody, test the selectivity of this antibody, compare with the well-established, well-accepted antibody that I just presented before. And our aim was also to re-examine the expression of SEKR1 in macrophages and monocytes. So first, we focused on the mouse, and we assessed the antibody that target the mouse SEKR1. So to do that, we used erythroid cell from the bone marrow of the mouse, and by flow cytometry is actually relatively easy to identify the cell as they co-express CD71 and TER119, as you can see here. And then we stain this cell with the antibody used by Hu et al. And the 6B7. And we did that, and we tested different concentrations for each antibody. And importantly, we used also erythroid cell from wild-type mouse, and so in red, and knockout mice, which then lack SEKR1. And you can immediately see that this antibody used by Hu et al. Equally stain the wild-type and the knockout erythroid cell. So demonstrating that this antibody is not specific for SEKR1. And in contrast, with 6B7, the signal was actually really clear in the wild type and completely absent in a knockout cell. So this also demonstrated that this antibody, the 6B7, is actually specific and recognized only SEKR1. So for the next experiment, we used this antibody because then it's a validated one. And we asked the question then, do macrophages express SEKR1? Because that was one of the main message from Wu et al. And again, by flow cytometry in a bone marrow, you can identify macrophages which express F480. And what we found is actually there were two subsets of macrophages. So, and especially the red one here, which express CD71 and TER119 markers, so two erythroid markers. And on top of that, you can see that these macrophages indeed also express SEKR1. So does it mean that Wu et al. Were right then? Do actually macrophages indeed express SEKR1? So to answer this question, then we turn to imaging flow cytometry, which is basically flow cytometry which take images of the cells. And what we found is actually this is not the case. Because we identify some cell complexes which consist of one macrophage here, F480, for instance, if you look at the lane 2. So you have one macrophage for F480, which is bound with two erythroid cells, which express CD71, TER119, and SEKR1. And it's the same for all the cells that we can see here. So this tells us that, and that's the conclusion of this experiment, that macrophages do not express SEKR1. Next, we assessed the specificity of antibodies that recognize the human SEKR1. So we did more or less the same experiments as I showed before, and you may wonder how then we will test the specificity of SEKR1 in human. Well, we are quite fortunate that there is a polymorphism for SEKR1 in the population. So the majority of people do express SEKR1 on red blood cell, but some people carry a genetic variant that leads to the lack of expression of SEKR1 specifically on erythroid cell. And this phenotype is called Duffy negative, and it's highly prevalent in individuals of African ancestry. So we used blood from Duffy positive and Duffy negative donor in order to test the specificity of the antibodies. And what you can see here from this experiment, so here again in the middle, that's the antibody used by Wu et al., and that's the well-established one, 2C3. So both antibodies recognize Duffy positive red blood cell and did not stain Duffy negative red blood cell. But you can also easily see that 2C3 is much more sensitive than this antibody. It's more sensitive because it better distinguishes Duffy positive and the Duffy negative red blood cell, and it does that at really low concentration. So for the next experiment, again, we used only 2C3 clone. And the next question was, do monocytes in human express SEKR1? So by flow cytometry, again, we identify monocytes which are CD14 positive cell, and we identify two populations. One population which do not express SEKR1, and again, a population which was positive for SEKR1. Now, you probably anticipate what we did next. We looked by imaging flow cytometry, and it turned out that monocytes do not express SEKR1. Again, this is a complex made of one monocyte and one red blood cell which do express SEKR1. So here you can see the overlay, a monocyte here and red blood cell, and the same for the three other images. So the conclusion of this experiment is monocytes do not express SEKR1. So overall, what we showed that by using validated antibody, we could demonstrate that neither macrophages nor monocytes express SEKR1. So The profile of expression I explained before at the beginning of the talk is actually the real one. It's only expressed by erythroid, venular endothelial cell, and Purkinje neuron in the cerebellum. So I don't know how long I have left. 5 minutes, perfect. So I told you that it took us nearly 4 years to publish this data. And I will go quickly, explain quickly why it was the case. So in 2016, Ur et al. Was published, suggesting that macrophages monocyte express SCKR1. 1 year later, the paper showing or demonstrating the generation of the 6B7 antibody was published. And the same year, we wrote to the editor of Stem Cell expressing our concern regarding Ur et al. Paper. And 6 months later, we received an answer. And they offer us to publish a Matters Arising article. The reason why they reply 6 months later is apparently they also received some complaint from other readers. So Matters Arising is a 4 figures, so we accepted the deal. At the time, we didn't have any data, so we worked on it for 2 years, generated all the data, wrote a manuscript, and we submitted so 2 years later. A month later, we received an email back from the editor that actually our manuscript does not fit anymore to Matters Arising. The reason why is to publish a Matters Arising paper, you need to repeat all the protocol using exactly the same technology, the same antibodies, and in the same condition, and demonstrate a different outcome. And by using another antibody, we didn't fit to this criteria. So what they offer instead is to write a letter, which is now 1 figure. So we had to condense our 4 figure into 1 figure. And they propose that to focus only on the mouse data, remove the human data. So we tried to complain, etc., didn't work. So we rewrote the manuscript as a letter with only mouse data. A few months later, we received the feedback from the reviewer. The first reviewer was really happy. Second reviewer, what about human data? So we, again, back discussion with the editor, put back the human data. And they allowed us to have an extra paragraph because, we have to put human data. So we work on it, we put even more data in 1 figure, including now mouse and human, send it back to the reviewer, or to the editor. Now she's happy. Problem is now Ur et al. Have the right to reply to our manuscript. And from here, we knew that then it will take also a while, because now the publication of a paper was dependent of the acceptance of the reply. And it didn't start very well, because a few months later, we received a letter from Ur et al., and it turned out that the reviewer didn't like it, mainly because the Ur et al. Used just more new, non-validated antibody to confirm their previous data. So we agreed that we will share the 6B7, however, we don't have the intellectual property. So to make a story short, it took us nearly more than a year to get both papers accepted and published in Cell Stem Cell. So my advice is, if you want to make it quick, probably it's not a good idea to contact the journal, just publish to another journal. If you really want to do that and contact the journal, maybe publish it first or put it in as a preprint in bioRxiv. So I showed you the dark side of my story. I would like to finish with a more positive note. And after this episode, this dark episode, I actually discovered that there was a community of scientists which were really aware of the problem of the non-validated antibody, and this can really affect and damage the science, and probably is part of the reproducibility crisis. And on social media, or also I discuss online with some of you, And so it was really, really interesting. I found people who were willing to share their experience with bad antibodies or non-validated antibodies. Some people who are trying to find solution for antibody validation. I also received offer to develop antibodies. So now I have a collaboration with someone in Dresden who is making an antibody against another atypical chemokine receptor. And so that was really an enriching experiment for me. And this is also an example so that's a tweet I made about Carl's and Peter's paper when it was published. You all know about this paper about antibody validation protocol, and it was published in eLife. And what happened is, as you maybe noticed, Caroline Bertozzi retweeted this post. She also reacted to it, and then she even made her own post about this issue. So for those who don't know, Caroline Bertozzi, she's a Nobel Prize. So it was really, really nice to see that even a Nobel laureate is concerned about antibody validation and is willing to inform the scientific community about this issue. So with this, I would like to thank the team and also collaborator in London and Harvard colleagues who made the 6B7 antibodies, and would like to thank you for your attention, and I'm happy to answer your question.